So the goal for today is going to sort of wrap up on DNA structure, wrap up on nucleic acid structures. Okay? And I'm also going to talk a little bit about separation via centrifuge. Okay? All right. So this is sort of where we left off on Friday last time. We talked about the Avery experiment, and we also talked about the Hershey-Chase experiment. Okay? So Avery did his experiment, you know, the one where you turn bacteria from non-pathogenic to pathogenic via just passing the DNA to the non-pathogenic strain, and they showed that you can make it into pathogenic. Hershey-Chase, do you guys remember what Hershey-Chase was? That's the thing where with phage you got the sulfur labeled and the phosphorus labeled, okay? And they were able to show that as your phage later on replicates, okay, they only observed the phosphor that was labeled. So it sort of showed that the DNA propagates, whereas proteins do not, okay? So you can see the years were 1944 and 1952. So it's around that time of the century where people were very interested in biochemistry, okay? People were very interested in the function of DNA, okay? So obviously you guys should know that Watson and Crick, they proposed the double helix model in 1953. Of course, they got a lot of help from all these people, and then they put it all together and said, okay, so this is what DNA structure looked like. And actually, it's not the structure that is so important, but it's rather the information that the structure can give you, okay? So in their model of DNA, they proposed that not only the structure of DNA, but more importantly, it's how DNA is copied and how that DNA can be copied and is used to reproduce DNA using a templated process, okay? So that's what we're going to talk about for the first 10 to 30 minutes, right? So obviously nowadays, you're just like taking this and be like, yeah, I mean, that's how it is, right? However, you have to imagine yourself 70 years ago that wasn't so obvious, okay? So DNA, so let's say we know that DNA is the thing that passes genetic information, okay? And then the question is, how is your DNA reproduced, okay? How is it copied, right? So we'll come back and talk about Meselson and Stahl's experiment in several slides, but right now, let's just give you an idea of what are the possibilities. So take a look on the slide at the figure on the right. Let's say you have a parental DNA. This is the sort of template, okay? You can have 3 types of reproduction or copying of your DNA, right? 3 possible types. You got the conservative, so meaning that after you reproduce your DNA, you still have the old DNA by itself and then all the other new DNAs by themselves, okay? You have the semi-conservative where essentially one strand of the DNA is used as a template, and then you make another strand that is the new DNA, okay? And you do that for both strands of the DNA, okay? Obviously now, because through your middle school, high school education, you know that DNA is copied this way, okay? You know that, okay? But back in the days, they didn't, right? And you can have a third, last possible scenario where you have a dispersive mechanism where the DNA just somehow magically copy themselves, and you got in one strand old, new, old, new, old. It's random, okay? The copying mechanism is random, okay? So again, 70 years ago, 80 years ago, people don't have a single clue as to what's the mechanism, okay? So that's what we're going to talk about right now. You know, think about what kind of tools, okay? Because you guys are future scientists. Think about what kind of tools that are available, and then how we can use those tools to show that DNA is one of these 3. Okay, so it's like a little role play. All right, so actually the Meselson and Stahl experiment, we'll talk about that in a couple of slides, but the method that they used, okay, was actually they achieved the identification of your DNA copying mechanism as semi-conservative, and it was achieved through the observation of how DNA density as the result of using different nitrogen isotopes. So we're going to talk about what this sentence means. Okay, so turns out they were able to do some experiment which they can separate labeled and unlabeled DNA, okay, using centrifuge. Okay, and the centrifuge has with it a cesium chloride density gradient. Okay, and cesium is a very heavy metal ion. Okay, so it has a atomic mass of 133, so it's pretty heavy compared to like sodium and calcium, those things, right? So since it's very heavy, and as you're centrifuging it, okay, so you spin it real hard, and because it's heavy, it's going to get pushed down to the bottom of the tube, okay? And as you're spinning and spinning and spinning, eventually you'll have a concentration gradient, because the longer you spin it, you have most of the cesium being at the very bottom of the tube, and the concentration sort of decreases towards the top of the tube, okay? All right, so this was done using a centrifuge, okay? And essentially you get to an equilibrium gradient, okay, where you have, let's say, low concentration of cesium on top, and here is high on the bottom. Okay, and because it's heavy, so it gets pushed all the way to the bottom. But then you're probably wondering, like, wait a minute, then how come it doesn't all of it get pushed down? Okay, because your centrifuge, you can control the force at which you apply your centrifuge, right? You can decide how fast you want to spin your centrifuge, okay, and that will determine, let's say, how much of the something is spin down. Okay, in this case, you don't spin all of it down because the force is not strong enough, so then you still have a gradient. Okay? With me so far? Right? You have a very heavy metal ion, okay, and a lot of it, and then you spin it real hard. Then you generate a concentration gradient based on, because cesium is really heavy. Okay? So the top of the tube is mostly water. Bottom of the tube contains a lot more of these heavy metal ions. Now, molecules with higher density than water, okay, let's say you put DNA, okay? DNA is heavier than water, right? If you put DNA into this tube and you start spinning it, okay, because it's higher density than water, so it's going to move down along the tube until the density of such molecule equals the density of your metal ion gradient, okay? So does that make sense? Because you're spinning it together, okay? And this is actually a very useful method to separate things based on their density, right? So in their experiment, they have labeled DNA using nitrogen 15 and unlabeled DNA using nitrogen 14. So if you spin it down, then what you'll notice is that the nitrogen 15 DNA is going to be towards the bottom, whereas the normal DNA will be on the top because they have different density. Make sense? Because, you know, N15 has one extra neutron, so it's heavier. Okay, but all the other electronic stuff are the same. All right, so any questions on how you generate a concentration gradient using cesium chloride? Cool. And how, because it's very heavy, so you can generate a gradient of density because this thing's heavy. So at the very bottom of the tube, the density is very high. On the very top, it's water, so density is low. Okay, in the middle, you have a gradient. Cool? All right. So very briefly, I want to talk a little bit about centrifugation, okay, because centrifugation is very important in all aspects of biological research, okay? Especially this thing called the differential centrifugation, right? So again, separation and purification is a key technique in all areas of chemistry and biology. Okay, so it's always about the ability for you to isolate something, because if you have a mixture of solutions or mixture of tissues, you don't know what you're studying, okay, because it's just a mixture. Okay, so for chemistry and biology, we always want to be able to isolate certain things. Okay, and centrifugation provides an easy method for separating some of the biological parts. Okay, for example, differential centrifugation applies different centrifugation speed to pellet, okay, to sort of make something into a pellet, different components of the cell. Okay, so you're spinning something. Okay, if you spin it very, very hard, everything comes down. If you spin it medium hard, only the super insoluble things will come down. Some of the insoluble things can still be in the middle. Okay, so this is a differential centrifugation. You centrifuge at different speed. Okay, this separation is achieved based on the size of the molecule, the size of the biomacromolecule, the parts of the cell. Okay, it's also based on the shape and the density of the particular stuff that you're treating. Okay, so let's take a look at this particular chart right here. Okay, the label says isolation of cell organelles. Okay, let's say I want to study a protein that's found in the mitochondria of a cell that's in your liver. Okay, and you're just like, okay, how do I get mitochondria out, right? Okay, so you can do this using differential centrifugation. Okay, so what you have is you got a liver. That guy is a liver. Okay, and you got some buffer. Okay, this buffer could be phosphate buffer or something, just helps to maintain the pH so that when you break the cell open, your enzymes don't like die right away. Okay, because you need to sort of maintain the environment at a specific pH. Okay, so then what you do is you chop up your liver, like you chop it off, like how you would do it in like liver soups and stuff like that. You chop it up, dump it into the buffer. Okay, and you're going to use a homogenizer, or just you homogenize it. Okay, so you're just like using a blender, and then you blend your liver into the buffer. Okay, so you have a liquefied liver. Okay, now what you can do then, you'll do some very basic, very rough filters. Okay, so you filter out anything that's not broken. Okay, so you filter out the things that are not broken. And then down here, that's sort of like your cells, like very small particle, your cell organelles, everything, proteins, whatever, everything's there. Okay. Then what you can do is start using centrifuge. Okay. So what you can do is you centrifuge it at about 600 G. Okay. So 600 G. Then you can remove the nucleus. Okay. So nucleus is pretty heavy. So nucleus will come down. Cytoskeleton, like your microtubules, those guys, they'll come down because they're very packed with proteins, okay, and stuff like that. Nucleus obviously packed with DNA. Okay, so these things will come back down. So let's say you're studying DNA or you're studying something about the nucleus. There you have it. You can take this guy out and then do stuff with it. Okay? And then what you can say is, nah, I don't want it. I take this, I do another centrifuge. Now I'm going to do at 15,000 G for 15 minutes or seconds. Okay? I think that's minutes. And then you spin it real hard, and then what you'll get is the mitochondria, the lysosomes, the peroxisomes, and if it's a plant cell, the chloroplast. Okay? So you can remove the organelles. Okay? The organelles are spun down. Then what do you have next will be, let's say you want to spin it again, okay, and then you can get out the plasma membrane, small vesicles, microsomal fractions, the ER fragments, okay, those type of things. Right, so the lipid stuff, you spin real hard, like really hard. You can even remove some larger proteins like viruses, ribosomes, macromolecules, that kind of stuff. Okay, and then whatever that's on top, you take out, and that will be your cytosol. Okay, anything that's soluble inside the cell. Okay, there you have it. Okay, so you can use centrifuge at different power. That will allow you to separate different parts of the sample. Okay, so this is something that maybe later on you guys will use as you're studying some kind of protein or some kind of a behavior in primary cell culture. All right? Now, a key thing here that I want to mention is how do you prepare these homogenates, okay? How do you break the cells open? Okay, you don't seriously use a blender, okay, because that's not efficient enough. So these samples are really first homogenized, okay, that is breaking the cell membrane. We pop the cell open, and these homogenates, the things that are, leaked out, are then fractioned using differential centrifugation. Okay, now the picture on the left shows you 4 common types of methods that we use to break cells. Right? For example, in my lab, I often or frequently use number 1, number 2, and number 4. Okay, but all of them are all used by different labs. Okay, so the first one is a sonication. The first one is called a sonicator. So it's this metal rod that vibrates super fast, okay? So it's like it vibrates so hard that the vibration, okay, the high frequency sound from the vibration is going to just like break your cell open, okay? So it's very violent. Okay? So obviously, when you're working with this particular instrument, it's better you do it in a little box, okay? Because if you let it vibrate in air, it actually will damage your ear, okay? So sonicator, super efficient, okay? Very, very useful. Most people actually prefer to use sonicator because sonicator is really easy to use and you don't have to do a lot of cleaning afterwards. Okay, it's just a rod, it's very easy to clean. The second one is using a mild detergent. Okay, because our goal is to break the cell wall open or break the cell membranes. Right, so then all you have to do is apply a mild detergent. You don't want to use something very strong like SDS because that's going to kill your protein as well. You want to use something mild, okay? And these mild detergents will be used to dissolve the cell membranes. Okay, you dissolve the cell membrane. Cool? And this is useful for a high throughput purpose. Okay, let's say today you want to process 300 samples. Okay, if you use sonicator, that's going to take a long time because imagine yourself with a sample, sonicate. Each sonicator takes about like 3 minutes to sonicate. Okay. Sonicate 3 minutes, you have 300 samples. Okay? Yeah, that's not going to work. Okay? So then, in this case, your detergent method becomes very efficient. Okay? Because you can add detergent, and adding detergent only takes you like 2 seconds. Okay? Per sample. Okay? So after you add detergent, all you have to do is spin it down, get the cell debris out, and then the soluble fraction will be your cell stuff, your proteins and DNA. Okay? Detergent, very useful. Limitation, it's not going to be able to process a large amount of cells, right? Because think about it, it's a mild detergent, so effect of it breaking your cell wall, it's not going to be 100%. Okay, sonicator can give you a very good efficiency, okay, with a lot of cells. So let's say if you want to have a very concentrated protein solution, you probably use sonicator, okay? Mechanical method to break it. If you just, I just want a little bit of my protein, detergent, okay? It's very easy to use. And you actually can buy this detergent from commercial vendors, so it's quite easy. The third type, this thing is called the French press, okay? Essentially, it's also just a mechanical way to break the cell, right? So what it's doing is you're gonna load the cell into a chamber up here, and you have a piston right here that sort of just presses the cell really, really hard, okay? And then here, you have a hole, okay? A very, very, very narrow hole, okay? Very narrow. And as you're pressing it really, really hard into a very, very narrow hole, your cell gets lysed. It pops it open, okay? So this is the French press. And you can also do a lot of cells using French press, okay? So it's very easy to use, yes, but it's annoying when you're trying to clean it, right? And French press is usually a very big machine. Right. I don't know if many labs still use French press nowadays, but my previous lab had it. Okay, so I never used it because I just thought it was annoying. Right. Number 4, this is essentially, it's sort of like it's similar to French press, but in a way, it's just grinding. Okay, so you have a glass tube kind of, and then you have this thing that grinds the cell. So you put your cell in the tube, and then you're just like grinding it real fast. And as cells are getting smooshed up, it leaks from the outside. So that's the lysate. So we got 4 common cell lysis methods, 3 of which are mechanical methods. Okay, so this guy, these are all sort of mechanical lysis. Okay, so based on like, just brute force, you like pop it open. Okay, so then let's say you pop it open. For example, you have a muscle cell, okay, muscle tissue. You homogenize it, okay, you break it, okay, you break it. And you got these tissue homogenates, okay. Then again, you have differential centrifugation, okay, like what we just saw in the previous slide, where you spin at different speed that can give you different organelles or different parts of the cell. All right, cool. After that, once you get all these like the cytosolic stuff, things that you don't really separate using centrifugation anymore, using differential centrifugation anymore, okay, because you spin it as fast as you could, okay. Now you can use this gradient centrifugation, okay, so then you can separate based on their density, okay, so use a sesamian chloride centrifugation gradient, and then you'll be able to separate, okay, or in this case you can use sucrose nowadays. Sucrose can do that too. You'll get like surface membranes, you'll get like other stuff, other dead cell debris. You'll get the heavy and the light sarcoplasmic reticulum fraction, etc. Okay, so you can apply different techniques together to get a finer separation. So differential centrifugation is after your cell is lysed, it's in your buffer. Okay, it's in your normal like phosphate buffer, so whatever buffer you use to lyse the cell. And then you're just spinning it at different speed. Different speed meaning different force, right? So you're separating based on size, shape, density. Okay, sure, no problem. So it's like the first level of separation, right? You're based on different speed. Now, after you applied a very strong force on it, and then you still cannot separate some of the parts inside, for example, your cytosol fraction. Okay, you want to have a finer separation, then you can use a gradient centrifugation. Okay, where the gradient centrifugation in this particular figure here, they're using sucrose. Okay, and the previous, of course, we use a heavy metal ion solution, but sucrose can do the same because sucrose has a different density than water. Okay, so you would mix these things with sucrose solution, and then you spin it. Okay, then what you can see is that you'll have a separation based on the component's density. It will go with the sucrose gradient. So it's just ways that help you to separate whatever that you're separating. All right, so let's come back to this Meselson and Stahl experiment, which shows how DNA replication is based on, it's a semi-conservative method, okay, or mechanism. As I mentioned, they're going to do this experiment using 2 different nitrogens. Okay, so obviously your DNA has nitrogenous base, so it has nitrogen, and we're going to have N14 and N15, 2 types of nitrogen. One is labeled, one is not labeled, essentially. Okay, heavy nitrogen and light nitrogen. All right. Now, before we go on, so this is actually a picture in your book. So if you have your book, you can take a look at it. But if you don't, on the left, this is pH 7. This is pH 12. What's the difference? pH 7, your DNA is double-stranded, intact. pH 12, they separate into single-stranded DNA. Okay, and obviously, what you can see is that under heavy, this is N15. Okay, so this is the heavy nitrogen. So if you grow your cell in heavy nitrogen, okay, like you just grow it in heavy nitrogen, the DNA that comes out obviously is going to incorporate the heavy nitrogen, which has a higher density. Okay? So now on the graph, this is density on the bottom, so it's obviously higher density towards the right, lower density towards the left. Okay? Cool. So regardless of whether or not it's single-stranded or double-stranded or single-stranded, you only get one type of DNA. Okay, of course, because you incubate it or you let your cell grow in the heavy nitrogen. Okay, so all of its DNA will be heavy. Now, if you grow your cell in the light nitrogen, N14, okay, then all of the DNA will have the light, okay, N14 nitrogen. Hence, you notice how the DNA that results from growing it with N14 has a lower density. Okay, so if you can't see the number, this guy is 1.724. This guy is 1.710. Okay, so it's lighter. Right? Cool. Now, so a simple experiment would be, what happens if I first grow my cell using N15? Okay, I grow my cell using N15, and then I all of a sudden switch it to N14. Okay, and look at what will be the DNA. Cool. Right? So in one generation, so this is one generation. Okay? What they observed was, I don't have a heavy, I don't have a light, I have a medium, right? Because this will be the heavy, this will be the light. I don't have light or heavy, I have medium, double-stranded DNA. Okay? Immediately, this tells you conservative model is out. Okay? Why? Because conservative model says, okay, after I replicate my DNA, my old DNA double-stranded and the new DNA double-stranded. Okay? So immediately you know that's not true. If it's conservative, then you will see this peak and this peak. You'll see two peaks. Make sense? Okay? We're good, right? So that will be conservative. So we know it's not conservative. And in second generation, in 2 generations, so I let my cell grow again, continue to grow in the light nitrogen for a little bit longer. Okay? And then I see my light DNA, but yet I still see my medium. Okay? That makes sense because the new DNA that's being made are all light now. Okay? That makes sense. Okay? You're thinking, okay, so it's not conservative. So what are my other 2 choices? Semi-conservative and dispersive, right? So I still cannot rule out my dispersive yet with this observation. Why? I can't, right? Because here in the first generation picture, it's a mix. So I don't know, like, is it a mix? One is all heavy, one light, or maybe this guy is 50 heavy, 50 light. This guy is also 50 heavy, 50 light. I have no idea. Okay? So then we're going to look at the single-stranded. Okay? So the single-stranded will tell us some information. Right? So if I then take this DNA, switch it to pH 12, it's going to separate into their single-stranded form. Okay? Then what I notice is I get very defined 2 peak. One light, one heavy. Okay? This tells me that it's not dispersive. Why? Because if it's dispersive, then each strand of the DNA will be a mix. Will be a mix of heavy and light. So then every strand will be like here or here or here. You'll have a mixture, right? If it's dispersive, you will have mixture of DNA that's in the middle. But the result shows I only get light or heavy after they're single-stranded. Okay, so this tells me that if I separate my DNA into their single-stranded form, okay, then all I get is a heavy strand, light strand. This tells me it's a semi-conservative mechanism for replication. Okay, does that make sense? Okay, so can I count on you guys if I ask you a question based on this and say, what would happen if it was a conservative mode of replication? Will you be able to draw for me what would you observe for pH 7 and pH 12? Okay? So this is something that hopefully you can get used to. Cool? So you should be able to answer that. What would happen if the DNA copying mechanism was dispersive? Okay, what would you see? Cool? All right, Meselson and Stahl experiment. So this tells you that it's semi-conservative. And together with Watson and Crick's model, it makes sense. All right, so that's that. Now, let's talk a little bit more about the chemical structure or the structural aspect of DNA. Okay, so for every base of DNA that we have, right, you have, this is oxygen, by the way. Here is your nitrogen base on the left, okay? And then you have this guy is C1, C2, C3, 4, 5, okay? Let's first take a look at the flexibility of your double-stranded DNA. Which bonds can move and which bonds cannot really move, right? So essentially, all of the bonds that are not part of the ring can freely rotate. Okay, I mean, that's very simple. It's not restricted into a specific circle, right? So you can rotate. That means this guy can rotate, that guy can rotate, so is this one, so is this one, this one, this one. They can all freely rotate. Okay, so if you have a nucleotide monophosphate, it's going to like rotate like crazy. It's very happy. Now, you have rotation of the sugar phosphate bonds. So bonds 1, 2, 3, 5, 6. So where are they? Okay. 1, 2, 3, 5, 6. So along the backbone of your DNA, you have rotation. No problem. Very happy. Okay. So they can get good rotation. You have also free rotation of bond 7, okay, which is this guy right here. The one that connects your C1 carbon to the nitrogen space, right here, this bond, that can also rotate. And the rotation generally have two forms. We have a syn structure or the anti structure. Okay? Syn meaning the face of the base is facing the sugar ring. Anti-structure meaning the face is facing the opposite direction. Obviously, which one is more stable? Anti is more stable, right? Because, you know, it's away from the sugar. You have less sterics, right? So it's happier that way. It makes sense, okay? And also, why does it make sense? Because we know this guy is C5, C5 with the phosphate, and this guy is C3, C3 hydroxyl. So we know that DNA backbone is like that. Right? So obviously your base is going to face inside, okay? Inside of the double helix. Okay? So this would be the outside of the double helix. All right. And then you have another slight movement, that is the different possible conformation of the deoxyribose ring. Okay? And you can have endo or exo. Okay, you're just like, what's the problem? Okay? Let's take a look. Okay? If we take, this is C5 and this is C1, okay, and if we say 4 and the oxygen, they don't move, so here they don't really move. So then you can have two possible things that can sort of stick out. Okay, so if you guys have in organic chemistry molecular models, you can actually build a 5-member ring, and you will notice how one of the carbon or one of the member of the ring always pop up or down. Okay, so if it pops up, we call that endo. If it pops down, we call that exo. Okay? So what you can have is, this is C2 endo. So on the C2 carbon, it's sort of going up. Okay? So it's like up. Right? Or you can have C3 go endo. Okay? Where C3 is up. Okay? So they're just 4 different conformations that your ring can have. Okay, so nothing too special, right? So now, DNA, even though we always say, you know, double helix, etc., turns out DNA has different forms. Okay, actually, DNA can adopt into, specifically, we say 3 different forms, but in reality, it's all 3 forms kind of happening together. It's just a population. You know, it's always a population of the DNA at a particular moment which form it's in. Okay, so it's not like your DNA is always in form B or always in form A. Okay, but most of the time we can say, okay, most of the time it's going to be form B. Okay, now the form that the Watson and Crick model came up with, that form was actually the B form DNA. Okay, and that is because the B form DNA is the predominant form in aqueous environment. Okay, so it's actually favored in aqueous environments. And of course, DNA is naturally found in aqueous environments, so DNA is usually going to be in its B form. Okay, I'll show you what A and B mean in just a second. Right? A form is observed in lower humidity. So let's say you somehow incubate your DNA in something that has lower water content, then it's going to be adopting to an A form structure. Okay, so A and B forms represent the major forms of polynucleotide secondary structure. All right, these things we call secondary because they have hydrogen bonding, they have 3D structures, so that's the secondary structure of DNA. Okay, so let's take a look at the table right here. Now, A form, B form, most common. They're both right-handed helix. Okay, so they're right-handed, so they twist upwards. So if you right-hand rule, right? Back in physics, did you guys learn that? Right-hand rule. So if you go this direction, the curvature is with your right hand, right? So if you go this direction, the curvature it goes this way. So that's a right-handed helix. Okay. Left-hand rule, obviously it's different, right? Because your hand is curving differently. All right. So A form and B form, they're all right-handed helix. That's cool. The diameter, meaning this thing, the diameter for A form is larger than B form. That's cool. We'll see just a minute what that actually looks like. And you have base pairs per helical turn, meaning in order to complete one turn, how many bases do you have? Okay, so remember in our last lecture we said something like 10, right? Turns out for B form it's actually 10.5. Okay, so it's actually a little bit off. It's a little bit off. Okay, but no problem. A form is 11 base pairs per turn. Okay, per turn meaning turn is where, let's say you have one base right here. The next time it comes back after circling, that's one turn. Okay, so how many base pairs required to make a complete circle? Okay, that's one turn. All right. You also have different base tilt normal to the helix axis. Okay, so if here is your axis, your bases will be tilt instead of a complete horizontal. Okay, it's tilt. Right? And the sugar conformation is a C3 endo, C2 endo, all right, et cetera, et cetera. Okay, so these are some of the basic characteristics between A form, B form, and Z form. Okay, now, obviously you're wondering how is that relevant to you guys, right? So let's take a look. All right. Most of the DNA that we study, or most of the DNA that we see in their double-stranded form exists in the B form. Okay, they have 10.5 bases per turn. Okay, so that's that. And if you take a look from the top-down view, okay, top-down view, the B form DNA is going to look like this, where in the middle, it's very packed. Okay, and then outside, you have a bunch of water that binds with the phosphates on your sugar backbone. Okay? Obviously, A form is different. You see how A form is not very crowded on the inside. Okay? We'll see that. All right? B form DNA is most stable in aqueous environment because your water can fill in the minor groove. Okay, let's take a look at this bottom left picture. Okay, what's the minor groove? What's the major groove? This part is the major groove, and this part between the two sugar backbones, this part is the minor groove. Okay, so here the minor grooves, you got all the phosphates phosphates on here. So then, since it's very charged up here around the backbone, your water fills right here. Okay? So you have water filling right there. Okay? And this hydrogen bonding stabilizes the B form. Okay? So that's why DNA in water is usually B form. Okay? Now, double-stranded RNA and RNA DNA hybrids usually happen in the A form. Why? Okay? Turns out B form is not favored for RNA structures because the 2' hydroxyl would get too close to the phosphate. Okay? So what does that mean? Okay? It means that on the sugar, okay, you're gonna have to pay attention to my drawings, okay? Let's say you have, this is gonna be C1, C2, C3, C4, and then C5 is inside. Okay, so C5 is smaller. Okay? So this is B form, right? Where's C2? C2 is right here. Okay? C2 is right there. This guy is the phosphate. Okay, so this thing is the phosphate. Okay? So since C2 is right here, phosphate is right there. For DNA, that's fine. Right? DNA doesn't have OH right here, so they don't like get too close. But if you have RNA, RNA will have an OH group coming out of this carbon, right? So you have an OH group sticking out. And that OH group sticking out, it will get too close to your phosphate, so the B form structure is not as favorable for RNAs. Does that make sense? Because if you look at the B form DNA ordering, your phosphate gets too close to C2. So if you have a hydroxyl group on C2, then it will cause some problems with structure hitting each other. So then RNA prefers to have A form rather than B form. Okay? So also, most polynucleotides are not straight helices, rather they're tilted because, you know, I mean, that's just how it is. I mean, it's chemistry. Like, you don't have a perfect helix going up and down. Okay? It's slightly tilted. All right. Now, what's also very important is that local sequences also have an effect on the local structure of the DNA. Therefore, even though I say, DNA is always B, RNA is always A, but all these A form and B form should only be thought as an average value because, in reality, it's not going to be super perfect, okay? Because a lot of C's, a lot of G's, it's going to have a different play into your secondary structures, okay? So, Zphone DNA is this weird thing where it's mostly found in polynucleotides with alternating purines, pyrimidines in each strand. So for example, if you have CG, 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 or these awkward sequences, then you can have Z form. What Z form is going to look like, very small in diameter, and it's going to be very long. Okay? It's small but long. Okay? That's the Z form DNA. Right, but you rarely see that. Right, it's not very common. Now, in Z form, the pyrimidines are always in the anti-form, and purines are always in the syn form. Okay, that's just something that people have noticed. Okay. All right. More notably, your Z form DNA has a left-handed helical structure. Okay, that's different from A and B form. Okay. And then the exact function of this form is still not certain, says your textbook. Okay, that's the Z form. So primarily you see A, B, Z, 3 form. All right. Now, your DNA or RNA, okay, can of course be single-stranded. Right? If it's not doing base pairing, it can be single-stranded. All right. And obviously, it's not just going to be a single strand like a linear line floating in the solution. It's going to have secondary structures. Okay, so these are the different conformations of single-stranded nucleic acids, right? What do you have? You have A, random coil. Random coils happen a lot, okay? Random means there's no particular structure and they're just, wandering around. So the random coil structure of the nature's single strands, okay? there is flexibility of rotation of residues, and there's no specific structure. So it's just like floating around in random coils. Of course, it's still going to be coils because it will have among themselves hydrogen bondings. Okay? You have B, stack-based structure. Okay? Stack-based structures, they adapted by non-self complementary single-stranded under native conditions. So essentially, they can still have with themselves some kind of complementary single strands and in their native conditions. Right? What that means is, if you look at figure B right here, so it's gonna roll themselves into a helical-like structures, which then each of the individual bases, okay, in the bases, can sort of pull the chain into a helix, but there's no significant hydrogen bonding between the bases with one another. Okay, so it looks like that. And then C, it's a hairpin structure. Okay, hairpin structure occurs when you have self-complementary sequences, meaning, let's say you have on a sequence, you have A, T, G, C, A, T, blah, 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 blah. And then you have, it's going to roll back up where the A can pair with A can pair with T, C goes with G, and this guy goes with that guy, T goes with A, it goes to T. So when it rolls on each other, you will have a structure that looks like this. Like that. Okay? So that's called a hairpin because it binds. Okay? It sort of can, fall on itself. So after it folds on itself, you can make this thing called the stem loop structure. Okay, stem loop meaning like that, where you have base pairing here. That's the stem. That's the loop. Okay, the inside the loop meaning the loop doesn't have complementary structures or complementary sequences. Okay, does that make sense? So single-stranded nucleic acids can adopt the following forms of structure. Obviously, RNA would do this a lot more because RNA tend to be single-stranded, and RNA can have secondary structures that looks like that. Okay? So these are just 3 different forms of structures that the single-stranded nucleic acids can adopt. But primarily, most of the times, what you'll see is actually a random coil because it really depends on the sequences of each different nucleotide in their sequence and then what kind of structures they will adopt. Which is why RNA is very interesting because it has all sorts of different kinds of structures. Okay, so that's that. Just to tell you that these structures exist. Okay. And there's a special type of hairpin called Cruciform. This is essentially nothing more than just a double hairpin, okay? And in order for this structure to form, the sequence must be palindromic, okay? And palindrome is just something that when you read it from the front towards the back, it looks the same as if you were to read it from the back towards the front. Okay, that's what palindrome means. So, for example, if you're going to fold into a cruciform, in this case, you would have something that looks like this, which is a palindromic sequence. Okay, so in this particular example, if you see how you got G, A, G, T, A, G, and then you got CTA, CTC. Then on the complementary base strand, it reads this way and it reads that way. It's the same. So then when you normally have base pairing between this guy, but as DNA is always moving, okay, DNA moves, and there's going to be after you break this guy into a single-stranded, what you can have is where this part folds with itself, okay, rather than folding with this guy, okay, such that you will have like this. So you're going, okay, and then immediately you fold this thing, okay, so such that you go up like that, okay, and because you have 2 of them, here you have base complementation, then they will still bind, no problem. Okay. However, in this case, the palindromic sequence sort of just sticks out, okay? That's called a cruciform. And of course, why is this useful? Because the ability to form these type of things will give your RNA a secondary structure, and that could affect, for example, translation. It could affect RNA protein binding, that kind of things, okay? So always having a 3D structure is essential to these purposes. So again, most RNAs are usually single-stranded, okay? Most of them will have self-complementary regions that allow them to form hairpin structures, okay? And some of them actually have very well-defined tertiary structures. And by tertiary structures, it means how they are able to form into a larger structure. So secondary is like the alpha, beta, Z structures, right? And cruciforms. But then tertiary is when you have a larger set of them together. That becomes the tertiary structure of your RNA. So if you were to go back to my earlier slides about how we have a RNA world, a possible RNA world, remember that? And how RNA could potentially serve as a catalyst to catalyze some reactions, okay? And then there was an example of a RNA catalyst, which we call the ribozymes, okay? And ribozymes sometimes have a very specific structure. That kind of structure is our tertiary structure, okay? So it's a bigger structure, okay? So these 3D structures have a lot of importance in their functions and roles inside of biology, okay? So I think this is actually done a while back, maybe 10-something years ago. So, you know, as technology is advancing, especially in our ability to compute, okay, a lot of people are interested in designing DNA into specific shapes, okay? And why is it that we can do this to DNA? Okay, because DNA folding or DNA binding, okay, it's very specific. Okay, DNA is always into their double-stranded nature. It's always folding to their double helix, okay? So if you use computations, you can sort of model out what kind of sequences that would be necessary to fold DNA into specific shapes, okay? And this thing is called the DNA origami, okay? You know, back then, you know, it's the ability that you can do these type of things that is making scientists very excited, right? Now, because you can have DNA having three-dimensional shapes, people actually are creating enzymes out of DNA, okay? That's interesting. Okay, so if you're interested in hearing more about that, I can tell you more about it. But, you know, DNA is nice enough so that we can predict what their structures will be if we give them a particular sequence. RNA is slightly harder because single-stranded RNA having that extra OH group, it can do a lot of additional hydrogen bond formation. So the structure is slightly harder to predict, okay? But people can do that as well, right? So here's a table of some common RNAs that you see inside biology. You can have RNA range from very, very small to very, very large. Okay, for example, some of the smaller ones are the tRNA, which stands for transfer RNA. These RNAs are used to load your ribosomes, okay, to load amino acids onto your ribosomes. So these are involved in translation. Okay? For example, if you have a tRNA for leucine, that means that it transfers leucine in a protein synthesis. Okay? So these things are small for recognition purposes, okay? Less than 100 base pairs. You also have ribosomal RNA. Okay? Ribosomal RNA are components of the ribosome. Okay? So if you take a look at E. coli, you have 3 different rRNAs. They're called the 5S rRNA, 16S rRNA, etc. Okay? And essentially, they're part of the ribosome. Why does your ribosome need to have RNA? Because ribosome decides when translation starts. Okay? So your ribosome can recognize DNA. Okay? So you must have some kind of a nucleotide component in your ribosome to recognize DNA, to recognize your tRNA, that kind of stuff, okay? In particular, 16S rRNA is very important in the field of getting the phylogeny of your strain. Okay, let's say today you isolated an unknown bacteria and you want to know what that bacteria is. Okay, let's say your 16S rRNA is a very useful thing for you to classify what organism is this. Okay, because your ribosomal RNAs are usually conserved to some extent. Okay, because all bacteria or all organisms have ribosomes. Ribosomes have ribosomal RNA. Okay, so all organisms have that. So then all you have to do is get the sequence of this thing, and then you see how far is it from E. coli, how far is it from other microorganisms. Okay, so you can use that. And then, of course, the most important type of RNA for our interest would be the messenger RNA, okay, because messenger RNA are the genes. Okay, so your proteins are made based off your mRNA, stands for messenger RNA, okay, and messenger RNA really varies in size. It depends on how big your protein is, okay. Typically, you go from somewhere like 600-700 to several thousand base pairs. Okay, that would be your mRNA. Okay, they code for an enzyme. Okay, so that's the messenger RNA. Once you have those, you can also have viral RNA. Those are RNA from a virus. Obviously, you guys know that some viruses, they don't have DNA. They only have RNA. Okay, so RNA-based viruses. Okay. So in RNA-based viruses or RNA viruses, okay, you also have single-stranded RNA virus, double-stranded RNA virus. Okay, if you guys are interested, I highly recommend you guys take a class called virology. Virology is very interesting. Okay, viral RNA, if it's RNA-based virus, that means that it's going to have to be able to replicate the entire virus from this RNA. So this RNA is usually larger. Okay. For example, you got polio. Okay, polio virus. Polio virus is about 7K. Okay, in length. And then you're probably wondering, is 7K large or small? What do you guys think? It's actually very small. Okay, it's very, very small. An average size gene is about 1K. Okay? It's about 1,000 bases in nucleotide. So that gives you about 300 amino acids. So that's like an average sized enzyme. Okay, so this is some numbers that will be useful for you guys later on. Like if somebody asks you, like how big is a normal enzyme or the average enzyme? So roughly speaking, a monomer of an enzyme, the average, like the most commonly seen enzymes, is probably around 300 to 400 amino acid residues. Okay, so that gives you about 1,000 to 1,200 in DNA sequence. Okay, so understanding that your polio RNA is only 7,000 or let's say 7,500. Okay, 7,500. It's pretty small. Okay, if you think of it, it probably only codes for some enzyme. Okay, so this is why I say if you guys are interested, virology is a very interesting class. Okay, there's this thing called the splicing. Actually, you guys know what splicing is. Okay. You have overlapping coding regions between two proteins. Okay? That's how these small viral things are able to package a lot of information into small segment of DNA or RNA. Okay? So again, if you guys are interested, definitely look into that. Yeah, so you have different kinds of RNA viruses, and usually they're not very large. Okay? Now, it wasn't known back then, but now it's increasingly becoming more aware of this, and it's more important. That is the microRNA. So microRNA is very small, usually like 10 to 20 bases. Okay, microRNA, or like around 20. Okay, so microRNAs, they happens in almost all eukaryotic organisms. Okay, what they do is they sort of control your genetic expression. Okay, they can bind to mRNA. That's one way. Okay, if you have a microRNA, a very small fragment, let's say you have messenger RNA supposed to make a protein, and somewhere here you bind a very small microRNA. or let's say this is the mRNA, you stop the translation. Okay, you block translation. So microRNA is quite important. It's involved in a lot of diseases. So if you're interested in studying the molecular mechanism of some common diseases, definitely look it up. Okay, look up microRNA. This is important, and you should know what this is. Okay, so just know the existence of the things. You will probably touch more on this when you get to molecular biology. But for biochemistry, my goal is for you to just hear these terms, just know their existence. Okay. Now, DNA. Okay, this is obviously very important. You know that DNA is very long. Okay, DNA is very long. DNA is quite long because it has to carry the information necessary to generate you an organism. Okay, so there's a lot of information stored in DNA. And because there's a lot of information stored in DNA, the space in which the DNA lives in, okay, is important. Okay, so we have to think about how to package your DNA in a space. Because otherwise, if you just let your DNA always in its B form, floating around in double helix, then you're going to occupy too much space. Okay, so turns out naturally DNAs will undergo this process called supercoiling. Supercoiling happens in bacteria and archaea because their DNAs are circular. Right, by the way, supercoiling happens to circular DNA. Okay, bacteria and archaea, since they have circular chromosomes and also plasmids, right? Plasmids are DNA that are independent of your chromosomes. They tend to be supercoiled inside of the organism that that they belong in. So these circular DNA are typically supercoiled in vivo to save space, right? So let's take a look. On the picture on the left here shows you the difference between a relaxed DNA and a supercoiled DNA. So immediately, by looking at this picture, what you should probably think of is that looks kind of like a rubber band and a rolled up rubber band. Rubber bands, it's a circular structure, right? And then you twisting it, twist it. Doesn't it kind of look like this? Okay, so turns out supercoiled DNA, let's say when you have a circular DNA, if you relax it, it's going to occupy a large amount of space. So when you supercoil it, you're rolling the DNA such that they fall on each other. Okay, that's supercoiling. Okay, so that they occupy a smaller amount of space. If you run your DNA on a gel, supercoiled DNA, because it's supercoiled, and because if it's supercoiled, it's going to be smaller in terms of size, okay, and the shape. So if you run it on an agarose gel, supercoiled DNA will run faster. Okay, so this is why if later on you guys start doing cloning, if you want to know the size of your plasmid, you're not going to take your plasmid directly run it on a gel. It's not accurate because the plasmid will be supercoiled. Okay, so if you want to know the size of your plasmid, you probably make a single cut. Make a cut, the DNA becomes linear, you run it on a gel, and then you see the real size of the DNA. Okay, so this is reflected in this particular picture on the right. You see how highly supercoiled DNA runs at the bottom, and then relaxed DNA runs on the top. Okay, so relaxed DNA, the shape is larger. Okay, of course the mass is the same. It's the same molecule of DNA, but shape makes a difference for DNA to travel through your agarose gel. Okay, so increasing in number, that's relaxation by topoisomerase. Topoisomerase is an enzyme that cuts your DNA, that relaxes the DNA. Okay, so that you make it more linear, and then you can see how gradually all the relaxed DNA becomes all the way on the top. Okay, so the shape becomes larger. So supercoiling is like this. So let's say you have a double-stranded linear DNA of 105 base pairs. So assuming the B form DNA, you have 10.5 base pairs per turn. And then you roll them into a little circle. Okay, that will give you roughly 10 turns because you have 105 base pairs. And since it's 10.5 base pairs per turn, so you have 10 turns. So this is sort of what we call twist, okay, in a circular. So if you take this guy and then immediately you seal this particular bond, then you have an unstrained circle, just like your rubber banding circle. Okay? The double-stranded circular DNA, unstrained, you have a linking number of 10. The twist is 10 turns, 10.5 bases per turn. You have width, W, which is 0. It indicates that you don't have a supercoil. What if you take this linear DNA, right? Okay, and you twist to the left one turn. Okay, you twist it one turn. Okay, but the length is still the same. Okay, and you link them together such that you only have 9 turns. If you only have 9 turns, then the base pairs per turn will become roughly 11.7. Okay, obviously that will be strained, right? Because your B form DNA wants to have 10.5 bases per turn. But if you unwind one turn, so then that means that in one particular turn you have more bases, so that you're twisting the bond angle in a way. Okay, so you have a strain in the circle, right? So that's not happy. As a result, what that guy is going to do, it's going to spontaneously pop into a supercoil. So all you have to do is then, for example, you like rotate half the circle into that. Then on the top, return to normal, where you got 10 turns, 10.5 bases per turn. So you reduced the amount of your strain, and then this is supercoiling. You start to turn your DNAs. Okay, so you let DNA wrap around itself. Okay, of course, this requires energy. So this is usually done by an enzyme that puts your DNA into turns, okay, into folded structures. Okay, so this is supercoiling, and it's used to save space. Let's take a look at some of the features, okay, that are useful for us as engineers or scientists. So let's first talk about the denaturation of double-stranded DNA to single-stranded. Okay, so DNA usually is double-stranded. However, if you apply enough heat to it, DNA is gonna, so with heat, DNA wants to vibrate. Okay, it wants to move around. You increase their average kinetic energy, so they want to move. And what happens is they acquired so much energy such that the hydrogen bond between the different between the complementary bases holding them together is no longer enough to keep them there, so then they break apart. This thing is called denaturation. Denaturation of DNA. Number one, when the native DNA is heated above a specific temperature, which we call the melting temperature, Tm, it is denatured, so it separates into single strands, okay? And the two random coil strands have higher entropy than the double helix, okay? So remember your delta G is equal to delta H minus T delta S. So if you increase, okay, if you increase the amount of temperature, then the delta S form, the delta S term dominates, right? So you want to have increased entropy. At low temperature, delta G is positive and denaturation of DNA is not favored. Okay? We're talking about this entire process. Okay? As temperature increases, minus T delta S overcomes the delta H, and this delta H, we're essentially talking about the hydrogen bond between bases. Okay? Okay. That's why you have a delta H there. Without touching the temperature, you have delta G equals delta H minus T delta S, right? The delta G for breaking DNA under room temperature is going to be positive because the delta H for doing that process is really positive, right? Because the delta H for DNA come together is very minus because of the hydrogen bond. So then the denaturation only happens if you give it enough temperature such that the minus T delta S term become large enough to overcome the hydrogen bonds of your bases, and then it breaks open. If you look at the graph on top here, we have increasing in temperature, we have On the left here is delta G for denaturation. And on the right here is fraction of denatured. Initially, and it's written by the red line, initially the fraction of denatured is at 0. Okay, because the red line is fraction of denatured. Initially it's at 0. Okay? And as the temperature increases, what you notice is that the delta G for denaturation, initially it's like very, very positive, and it's going to drop real quick until it gets to 0, okay, when delta G is equal to 0. And then your fraction denatured obviously is going to be 0.5. When delta G is 0, meaning half is in equilibrium, right? Half denatured, half not denatured. And then as the delta G becomes really negative, that means that you have a driving force for denaturation to occur. So then eventually it gets to all denatured. So why is this actually particularly important? In addition to that, later you'll see, it gives us the ability to do PCR. But what you can do is double-stranded DNA and single-stranded DNA, they have a difference in absorption. Okay, so they respond to light differently. It makes sense, right? Because when DNA is together, it's stabilized between the bases. Okay, so the amount of light it absorbs is going to be less than the amount of light that the denatured single-stranded DNA can absorb. Okay, so absorption spectra of native and denatured DNA show that the native DNA absorbs less light than the denatured DNA. Okay, that's cool, right? So this thing called the hyperchromicity of double-stranded DNA can be used to distinguish between native and denatured forms. This actually gives us an experimental advantage in determining the TM of your particular DNA. How do I do that? So what I can do is I look at wavelength at 260 nanometers, and essentially, let's say initially it's equal to 1. Okay, I just adjust it so that the beginning absorbance is equal to 1. Okay, and then I start to heat up my sample. Okay, and usually heating does not affect your absorbance until when you get close to TM, you see a sharp increase in absorbance and then it stops. The place where the slope changes the most, obviously, okay, you have an inflection point, then this is the TM of the particular DNA that you're working with. So you can use this method using the difference between light absorbing capability to identify the TM of your DNA. Now you're probably wondering why is that important? Well, it depends on your application. PCR, you have to know the TM of your primers. For some biosensing applications, you probably have to know at which particular temperature your DNA breaks, etc., etc. Okay, so there are different times that you will have to know the TM of your DNA. So this is a method that you can use to determine it. Because otherwise, how else would you determine the TM of your particular DNA? Right? You can only calculate, but that gives you a experimental method to look at the TM value for your DNA. Now, TM increases with the amount of C's and G's you have in your sequence, obviously, because the more GCs a DNA molecule has, the more energy is required to separate the double strands. And that is because GCs have more hydrogen bondings, right? Remember, we always draw G, 3 hydrogen bonds to C, A is only 2 hydrogen bonds to T. So if your DNA has high amount of G's and C's, then the TM is going to be pretty high. If you were to look at a guanine plus cytosine, so GC, we usually call this GC content. Okay, GC content. And then this is the melting temperature. You can go from 0 to 100, and obviously you see how the TM increases with the GC content. Okay, so GC content of 40 gives you a TM of 80 something. GC content of 60 gives you a TM of 90 something. Right, so the more G's and C's you have, higher the TM. So for the remaining 10 minutes or so, I want to talk a little bit about recombinant DNA techniques, because probably as a biotech major, this is something that you guys should care about. So recombinant DNA techniques are used to study nucleic acids and their transcriptions and translations into proteins. Okay, so of course, this is only a very brief introduction. You'll hear more about this when you get to molecular genetics or molecular biology, or if you decided that you're going to do a undergraduate research in a research lab, for example. My lab, we use probably all of this, okay? All right, so some recombinant DNA techniques include gene cloning, okay, because you need to be able to clone a gene in order to study the gene. Chemical synthesis of oligonucleotides for use as primers, sure, because how else do you have primers to do PCRs, etc. You have DNA sequence analysis, sequencing. Okay, that's important because we need to know what's the sequence of the gene that we're working with. You can do site-directed immunogenesis. We can evolve proteins. Okay, so I actually do this in my lab a lot, where we have to generate non-natural enzymes for a particular purpose. PCR, you know, homologous recombination, is a method in which we can deliver genes into an organism. Some applications for recombinant DNA techniques are, we have recombinant protein production, for example, insulin. Okay, we will talk about insulin when we talk about protein secondary structures. Insulin is the first real successful example of biotechnology, because insulin previously was harvested from cows and stuff, so it was too expensive. And insulin, you guys know what it's for, right? We'll talk about that. Growth hormones and vaccines. Okay, so some genetic defects require people to take growth hormones because they don't make them themselves. So we can make growth hormones. Same with vaccines. Right? A lot of vaccines are based on recombinant proteins. You also have genetically modified crops. I know most people would not like that, but believe it or not, you're eating genetically modified food every day. Okay, that's just how it is. And a lot of modifications are very beneficial for the economy. Okay, not necessarily beneficial for your health, but at least beneficial for the economy. For example, you can have insects and herbicide-resistant plants. You can have cold and drought-resistant plants. And how do you generate those things? You probably clone a insect gene into plants, things like that. Okay, although a lot of people don't like it. You have other things like synthetic biology or synthetic biological functions, like for example what I do in my lab, we do biofuel and biochemical production. Some other people are interested in biosensing. Can we use E. coli to sense the presence of another harmful bacteria? People are looking into that. We use recombinant DNA technology or techniques to study the specific function of genes and or proteins. For example, we can purify human enzyme using E. coli, and then after we purify it, we study it. You can't do that unless you have recombinant DNA techniques. Okay? Otherwise, how do you get human proteins? From human body? No one's going to let you harvest their proteins. Okay? So obviously you have to clone it from genes, put it into E. coli, and then get a lot of that protein. And many more applications involved in biotechnology, agriculture, medicine, and research. Yeah. How could it be bad for your health? That's up to debate. Okay, so I personally think that if it's genetically modified crops, so it's just DNA. But some people would argue that what if your DNA is not completely digested and somehow your body absorbed that DNA and go into your cell? Is that impossible? I wouldn't say impossible, but the chances of that happening, I think is pretty low. Okay, so that's one thing. Another thing could be allergy. Okay, for example, if I make a plant resistant to insects, that probably means that I expressed some kind of a foreign protein in this plant. But what if today your consumer is allergic to this particular protein? So people could be allergic to proteins, and this person didn't know. And then he ate this, and then he got an allergic reaction. that could be bad. Depends on how bad the allergy is, right? So anything that's non-natural is undergoing a lot of debate because it's not natural, humans have not evolved to coexist with this as a food. For example, some genetically modified crops may have proteins from like crazy insects that we never eat, right? I mean, usually people don't eat insects anyways. But, after we do this, what kind of effect do we have? Immediate effect? Maybe no, but what about long-term effects? So currently that's the criticism that genetically modified crops are facing. That's the thing. But, in my personal opinion, if it's DNA, it's DNA, right? Economy is important, right? Because without advanced biological research, agriculture could be a limiting step in feeding all these people on Earth. There's a lot of good things that you can do with recombinant techniques. However, a lot of scary things too. Creating recombinant DNA is actually a very simple process. DNA can be manipulated through amplification and then through this cut and paste technique. Right? So this is to any molecular biolabs, like for example, in my lab, this is like very routine. You will first amplify your target DNA, okay, using PCR. And very briefly, the way PCR works is that you first synthesize these primers. Primers are short oligonucleotides, usually about 20 base pairs. And you synthesize them. You have to have a forward primer and a reverse primer, such that, let's say I want to amplify this particular region. I would take my template DNA. This template DNA could be from whatever organism's DNA that you isolate. You isolate their DNA, and I know this is the chunk that I want to make more, I want to amplify. Of course, before you can amplify it, you need to know their sequence. Okay, so genetic sequencing is also very important, or genomic sequencing rather. The first step in PCR is you let it denature at high temperature, typically 95 to 98 degrees. You let the template denature, and then you cool it down to roughly 50 to 60 degrees Celsius, such that your primers, because you designed it to complement it to the template, they're going to bind to your template. Of course, it is also likely where your template just bind again, right? But since we can put a lot of the primer, so the chance of primer binding to the template is very high. And then I adjust the temperature to usually 68 to 72 degrees. This is the temperature for thermostable DNA polymerase, okay? And it's going to do polymerization, so it will make you more and more DNA. Typically, any PCR experiment that we do, or PCR procedure, I don't even call it experiment now, essentially by 30th cycle, so this is, you can repeat, right? So denature, anneal, extend, and then you made DNA, right? And then you denature again, you do this whole process again. So repeatedly, if you repeat this 30 times, you can get about 2 to the 31 power in terms of copies. So that's roughly 2 billion copies. Okay, so that's a huge amount of DNA. So this is used to amplify DNA. And by amplifying or amplification, I just mean that we're making more and more, physically, more and more DNA of this particular sequence. The reason why we're making more and more of this particular DNA is because in order to manipulate this DNA, you have to have a sufficient concentration of it. You cannot just have one molecule and then you do stuff. No, you have to have a lot of DNA in order to do stuff to it. Okay? So that's that. Subsequently, after you amplify the DNA like this, then you can do recombinant DNA, meaning the cut and paste. Okay, so for example, I want to make my E. coli express a particular protein. Okay, then E. coli have plasmids that I can use. All I gotta do is cut my plasmid the same way that I cut my gene. Then I can ligate them together, transform them to E. coli, for example, and then we can use E. coli to overproduce your protein. Okay, so this is sort of the recombinant DNA for dummies, okay? So this is very, very simple. It's very, very simple. To do a complete cloning from primer to finishing the plasmid, it takes about like a week. That's it. Okay. And nowadays, a lot of companies doing synthetic biology, so synthetic bio companies, actually have complete automated systems using robotic arms, okay, colony pickers, so they can generate probably 10,000 plasmids in 1 week. Okay, so they can do very fast. So you can imagine where we can do very quick, high throughput generation of different gene variants of the proteins that you want to study.